Hi, hello everyone. My name is José Carlos Segovia, and today we are gonna talk about the treatment of pyruvate kinase deficiency by gene therapy. Let's go to share my screen. Why pyruvate kinase deficiency is a good candidate for gene therapy? Okay, because the PKD is a monogenic genetic disease, can be cured by allogeneic bone marrow transplantation. That means that healthy hematopoietic cells can cure the disease, and also because we know the gene responsible for the disease, and the gene has been cloned and can be introduced in gene therapy tools. There are many gene therapy clinical trials based on the hematopoietic system ongoing, and here in this slide I have reflected most of them, and at least there are two of them: the treatment of adenosine desaminase immunodeficiency and the treatment of beta thalassemia that have been already approved as medicines for the treatment of these patients in the clinics. Here is the pyruvate kinase deficiency, which is it's going to be the third erythroid disease that is going to be treated using gene therapy addition. So, how does ex vivo gene therapy work, which is the type of gene therapy that we could apply for the treatment of PKD? So first, we obtain cells. Cells are extracted from the patient. Some cells, the target cells. In our case, the target cells will be the hematopoietic stem cells. We will deep on that later on. On another hand, there is a virus that can be modified to maintain their ability to introduce genetic material into the cells, but that the virus is this virus is removed from any damaged proteins, and also it cannot be reproduced in the cells. So once this virus has been modified, the gene of interest, in our case, the gene responsible for the expression of the pyruvate kinase enzyme, which is the deficiency in PKD, is introduced in this virus, and then these are called now lentiviral vectors, because the virus that we are going to use is an antivirus. We will see it later on, and it's a vector because we only use this virus to vectorize the genetic material into the cells. Then the cells are put in contact with the viral vectors, and then the viral vector will introduce the genetic material into these cells. These cells are already mentioned as corrected cells or cured cells, and these cells can be now transplanted again and be infused again in the patient, where they will start their function, and they will, in principle, restore the deficiency and cure the disease. So what are the tools that are needed? As I mentioned, we need some viral vectors, and in our case, we develop the lentivirus to develop a lentiviral vector for the transduction of the damaged cells. Why lentiviral vectors? Because these are easy to manipulate. The essential viral proteins can be provided in trans. That means that we can generate these viral vectors in a way that are not infectious, that cannot reproduce into the cells. They have a space enough to allocate the PK gene. And the most important thing, they can integrate into the host genome. That means that if we transduce one cell, these cells and all the daughter cells, all the cells that 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 are generated from these transduced cells are going to carry also the copy of the corrected gene. A second tool that was very important for us was the availability of a Pyruvate Kinase Deficiency mouse model, animal model, in our case, a mouse model. This mouse model reflects almost perfectly the disease that we can detect in humans. These mice are anemic, as you can see here. They have less erythrocytes, they have less hemoglobin, they have splenomegaly, as you can see, and they have also a clear reticulocytosis in the blood. So with this animal model, we were able to test if the developed tools could work as the therapeutic tool. For that, I have the opportunity to work with these three people that helped me in the development of these preclinical studies to test the efficacy of the lentiviral vectors developed for the treatment of PKD. We first tested if gene-corrected cells were efficient or sufficient to restore normal erythropoiesis. For that, we took hematopoietic cells from these mice deficient for the gene. We purify hematopoietic stem cells because are the cells that are responsible for the generation of all the cells, including the erythrocytes. And we transduce those cells with two different vectors. One just expressing non-important protein, just as a control vector, and the other vector that carries the healthy RPK gene. Those cells were then transplanted into, again, deficient animals, and we follow the number of cells transduced with the virus, as well as the erythroid parameters, to see if this transduction was good enough to restore the normal hematopoiesis, the normal erythropoiesis in these treated animals. For that, we tested different doses of virus to see what could be the threshold of corrected cells to cure the disease. Here we can see the different parameters that we analyze in the blood, including reticulocytes, hematocrit, hemoglobin, and erythrocyte counts. And as you can see, when we use an MOI, which means multiplicity of infection, or the number of viruses per cell, close to 0.3%, that means 30%, approximately 30% of cells corrected. Once we reach this amount of corrected cells, we were able to exit the deficient situation and enter into a healthy, normal situation. Similarly, for the erythrocyte count, for example, reaching 30% of corrected cells, 0.3 viral copy numbers per cell, we enter into normal values of erythrocyte counts as well as hemoglobin. At the end of the experiment, we also analyzed the size of the spleen of the different groups of animals, and again, as you can see, we reached normal levels when we were above 30% of corrected cells, between 0.3 to 0.6 vector copy number per cell. With these results, we could define that in a range of 30% of corrected cells with our lentiviral vectors would be good enough to restore, to cure the disease. We also analyze many other hematological parameters, like the erythrocyte half-life, the levels of erythropoietin, and also the erythroid differentiation, and we observe in all cases a clear restoration of normal erythropoiesis, not only in hematological parameters, but also in the organs. The spleen weight was recovered, as well as the problems related to iron deposits. Also, the liver was clear from iron deposits approximately when we analyze these animals 1 year after the injection of the corrected cells. We were also very aware about the safety of the trial, and we performed also preclinical studies of safety, and we did a biodistribution study of the medicinal product, which means where the corrected cells that have been transfused, where those cells go. And as you can see in this graph, we only were able to observe corrected cells in the hematopoietic organs, the peripheral blood, the bone marrow, the spleen, and we didn't see any signal of these corrected cells, of these injected cells in other organs, meaning that our cells were going to the correct place, to the bone marrow, and from the bone marrow, the hematopoietic stem cells were producing healthy erythrocytes moving to the blood and restoring the disease. With all this information, we prepare all the documentation, and in 2014, the European Medical Agency gave us the approval as orphan drug for this potential medicinal product, which means the autologous cells, the cells from the patient, in this case from the diseased animal, transduced with this lentiviral vector and correct. And this orphan drug designation was also obtained in the States by the FDA in 2016. So if we take a look to the development of a gene therapy strategy, we have these, let's say, 6 different steps, and the 3 first steps were accomplished up to this point in our institutes, in our academic facilities in the CIEMAT, Ciberer, and Fundación Jiménez Díaz institutes in Spain. But from them, we were lucky to get in contact with Rocket Pharma, who decided to join the journey, the trip, and move to the establishment of a clinical trial to test if this medicinal product could be applied in humans. With the help then of many people that I have reflected here, and also with the help of Rocket, we end up in the development of a gene therapy clinical trial for the treatment of pyruvate kinase deficiency. What are the main objectives of this trial? Of course, the main objective of the trial of this, in this case, of a trial phase 1, almost phase 1 trial, is first safety, is to demonstrate that the corrected cells, the cells, the autologous cells coming from the patient and transduced and corrected with these lentiviral vectors were not harmful for the patient, and evaluate the achievement of the engraftment of the cells into the patient, and also in the avoidance of any adverse effects. But also we want to know also if there is some kind of efficacy. So our secondary objectives are focused on the recovery of the anemia, on the recovery of all the pathological symptoms of the patients by the potential decrease or elimination of transfusion requirements, the reduction of hemolysis, and the functional recovery of corrected erythrocytes. How it's going to be, how is the clinical trial established? So first, we are going to obtain the hematopoietic stem cells from the patients. And for that, we will treat the patient with some drugs. Those drugs allow the movement of the hematopoietic stem cells from the bone marrow to the periphery. And then from the periphery, we can harvest those hematopoietic stem cells, purify the hematopoietic stem cells from the rest of the cells that are harvested, because it's like an heterogeneous population obtained. And then these cells, these hematopoietic stem cells will be put in contact with the viral vector that will introduce the corrected gene into these hematopoietic stem cells. Then those cells will be frozen and those corrected cells to do different studies. And later on, the cells will be thawed, and just after the hematopoietic conditioning of the patient, that means myeloablation, the elimination of all the hematopoietic cells from the patient, those thawed cells will be infused into the patient, and the patient will be followed for the recovery of normal hematopoiesis and the recovery of the disease. So why do we need hematopoietic stem cells for the correction? Why hematopoietic stem cells are the target of the correction? Okay, although we know that the main problem of the PKD patients is the anemia, that means the loss of erythrocytes. The normal erythrocytes have a half-life of approximately 90 to 120 days. And those cells need to be replenished every day because of this short half-life. Who are the cells that are responsible for the replenishment of those erythroid cells? Those are the hematopoietic stem cells. Hematopoietic stem cells lives all lifelong in the patient, generates new hematopoietic stem cells to maintain this population, and also its main job is to produce all the cells, all the blood cells that we have in the periphery, including lymphocytes, granulocytes, neutrophils, and of course also erythrocytes. So if we target hematopoietic stem cells and we are able to infuse those cells and engraft those cells into the patient, only one gene therapy treatment would be enough to cure the disease for the rest of the life of the patient. Why do we need stem cell mobilization? We need this mobilization to facilitate the recovery of these hematopoietic stem cells. Earlier, before the known of those drugs, the way to obtain stem cell was by stem cell aspiration, which requires anesthesia and is more painful treatment. With this treatment, with the hematopoietic stem cell mobilization, we can just get the cells from the periphery, from a blood catheter, and is more or less like a blood donation. So we need, as I mentioned, to allocate hematopoietic stem cells from the bone marrow to the periphery, and as I mentioned, we can easily collect them while they are circulating in the periphery. Why do we need freezing? We need freezing to assure and to have time to test the quality of the product. We need, before putting the product in the patients, we want to know if the product has been properly corrected, have the enough amount of corrected cells, enough amount of cells transduced and expressing the healthy gene. And also we need to know the viability of those cells, as well as the sterility and all the safety controls required to assure that infusion of the cells is not going to be harmful because of the procedure. And then why do we need, and also to give time, sorry, and also to give time for the conditioning, because the conditioning requires some time to get approximately 4 to 5 days. So finally, why we need conditioning, why we need myeloablation? Well, although the erythrocytes are the erythrocytes coming from the endogenous hematopoietic stem cells from the patient are defective, and the corrected cells and the corrected hematopoietic stem cells are going to produce healthy normal erythrocytes. The properties of both hematopoietic stem cells, the endogenous ones and the corrected ones, are similar. That means that there is no any advantage in the hematopoietic stem cells to be allocated in the bone marrow. So we need to make a space. We need to facilitate the engraftment of these corrected cells in the in the bone marrow of the patient, and that's why we need this hematopoietic conditioning to eliminate the diseased cells that can still be there and make space for the new healthy corrected hematopoietic stem cells coming from the protocol. Who are the patients that are going to be eligible for being enrolled in the trial? It's in general view that there are like 4 different groups of PKD patients, depending mainly on the severity of the disease. And in these first studies, we are going to target just the more severe ones, those patients that even after splenectomy, they still require regular transfusions for their life, which means that they are going to be requiring bone marrow transfusions for the rest of their lives. This is our target patient. And how is the scheme of the global clinical trial? The patients will be treated in the clinical sites. It's a global clinical trial that is performed in Europe as well as in the States. And in Europe, the clinical sites are the Fundación Jiménez Díaz Hospital for Adults in Madrid, Spain, and Hospital Niño Jesús for Pediatrics also in Madrid, Spain. And in the States, adults and pediatric patients are going to be treated in the Stanford Medical Clinic University. In these hospitals, the blood collection, the treatment with the hematopoietic factors and the blood collection will perform in those patients, in those hospitals, and the cells will be shipped overnight to a cell manufacturing platform, one in the States and other one in Europe, where the cells are going to be manipulated and corrected similarly and following standardized protocols and homogeneous protocols. So there, the cells will be purified and transduced with the lentiviral vectors that are going to be the same lentiviral vector production in both sites, and the cells will be will be cryopreserved. Once the cells are analyzed and the product is demonstrated that is good for their infusion, the cells are shipped frozen again to the hospital, where the patient will be called again to the hospital, will be conditioned, and the cells will be infused, and the patient will be monitored there for the recovery and for the potential cure of the disease. As I mentioned, the global clinical trial sites are going to be in Madrid, in Europe, and in California, in the Stanford University Hospital in the States. And the chronology of the patient enrollment is here. Actually, the clinical trial started in April this year, and the first patient has been already infused, and the first data of safety, potential first steps of efficacy will be disclosed by the end of this year, by December of this year. New patients are being enrolled, and scheme is reflected in the slide, where we are going to treat 2 adult patients, from 18 years old and above. Later on, we will treat 2 pediatric patients between 12 to 17 years old, and then we will move to more younger pediatric patients from 8 to 11 years old. Always patients that after splenectomy remain transfusion dependent. Finally, I would like to speak with you about the patient support that we are having. Some years ago, we contacted with this website and with this Pyruvate Kinase Deficiency group, and they asked us how can we help to the development or to move on in the gene therapy clinical trial. So the webmaster of this page just performed a short survey where they asked the relationship with PKD and if they would be interested in this trial. And it was very, very amazing for us to see that only just in 2 weeks we received more than 800 different answers, answers coming from many, many different countries around the world. And for us, also very important was to see that 37 real PKD patients answered this in this survey, and that many other people involved in the or related or affected by the disease, like parents or family members or even friends of the patients, clearly support the development of this clinical trial of this gene therapy treatment for PKD that is now ongoing, and that we will have results, hopefully good results, very soon. So in summary, we can say that hematopoietic stem cells corrected with these RPG expressing antiviral vectors restore the normal erythroid function and compensate the hemolytic anemia in a mouse model of PKD, and this was designated as orphan drug by the European and the American regulators. Human hematopoietic stem progenitors can be efficiently transduced with GMP antiviral vectors, and the gene therapy clinical trial for PKD has been started, and the first cohort of adult patients is ongoing. So finally, just I would like to acknowledge all the people that has been involved in this work from the very beginning, starting from Nestor Meza and Maria Garcia Gomez, that were the starting works, the starting doctoral thesis focused on the development of this gene therapy strategy for PKD. all the group at CIEMAT, Ciberer, Fundación Jiménez Díaz involved in the work about gene therapy for PKD. Of course, thanks to the doctors and the clinicians that have been involved and are working hard in this disease. Julián Sevilla as the principal investigator of the pediatric patients in Madrid, Spain. José Luis López Lorenzo and Lucía Llanos as principal investigators in Fundación Jiménez Díaz for the adult patients in Europe, and Amy Shaw as the principal investigator for PKD patients in Stanford University. And of course, my big acknowledgements for people from Rocket, especially from Jonathan Swarth, Kinari Patel, and Gorazio, who believe in our strategy and put a lot of efforts and a lot of work in putting this in a real clinical trial as it is moving nowadays. And of course, all the collaborators and researchers and doctors and all the patients and all the healthy donors that have provided us with blood and bone marrow samples for the test of the different tools and for the setup of this gene therapy protocol. Thank you all for being here for this almost half an hour discussing and talking about the treatment of pyruvate kinase deficiency by gene therapy drugs. Thanks a lot for your attention.